So let's move to cell characterization and comparability. And one of the exciting things that's happened is in the last few years is really the evolution of regulations. And I thought we'd talk a little bit about that. Is the efficacy and the products driving the regulatory changes, or are the regulatory changes kind of driving the products? Because there's a lot of change happening there. And I think people bucket regulatory, reimbursement, and manufacturing, but they're really all one. They have to be kind of considered together. And comparability and cell characterization is really one of the binders of all those things. But any comments on the evolution in the regulatory approval process around the world?
I mean, I think from my perspective, from an academic perspective, I think what's been very reassuring in this space is that actually the regulators have seen the pace of change. They've seen the need to integrate early on with companies, with academic group, with clinicians. And actually, I think there's been a good dialogue between the regulators. And I think, you've had, CDMOs like like Hitachi and others who have been informing the regulators in this space. And I think that dialogue has been critical. And I think that helps when you have these national initiatives such as CCRM. In the UK, we have the Cell and Gene Therapy Catapult. These are national initiatives which are now established to facilitate the standardization, facilitate the regulation, and to try and develop the reimbursement space as well. And I think it's great to see the competition in Japan. I mean, the government initiatives to change the regulation and reimbursement space, I think, shook the industry up, and I think that's created this change and allows us to start to compete more on a global scale.
Bob, you work in Japan. Any comments on what are the implications of that change in Japan?
I would just agree with Qasim. I mean, they shook things up, right? They took, it was actually an economic decision more than anything. They're looking for the next
the stem cell nation. They really wanted to
1,000 people a day.
Yeah.
The population of Japan is decreasing by 1,000 people a day. It's an aging society. The most of the industry there is older industry that is now on razor edge of margins and the like, and it's very difficult for the companies to stay. The economy struggles, and so the Ministry of Economic and Trade recognized, based on Yamanaka's work with iPS cells, recognized this is the next frontier, and put out I don't know what the right word is, edict or order. A mandate that the PMDA and others come up with some mechanism to facilitate the growth of the regenerative medicine industry in Asia. They just did it. And I find that that's part of my experience with Hitachi too. That's just kind of how things happen. They talk and then boom, things just happen. And so he's like, wow, that was pretty quick. They didn't get stuck in some of the stuff that our regulators got stuck in.
Yeah.
Because they had a government mandate. Here in this, in the U.S., in the U.S., the FDA has been an incredible partner since the early, when they didn't know what these products were. And the first product that we brought was a device. It was the Dermagraft product, and that got approved as a device, not as a cell product, because there was no regulatory framework. But they've been collaborative, they've been helpful, they've been tremendous. And the same we find in Europe. The difference is that in those two markets, and maybe perhaps here too, well, this is not as litigious a society, but certainly the U.S. is, that it's a very risk-averse group, FDA, and they're afraid to make I think they understand the profile here. And I don't think they particularly are afraid of it, but that's just the style. Having broken that mold now in Japan, the rest of the world has said, hey, we can do this too.
And especially with products that are showing efficacy.
Absolutely.
Let's get them to the patient.
They've always made that easy, I think, right?
Yeah, okay.
The challenge we've had with regulatory agencies more is on the side of industry and academia that didn't understand the regulatory environment. And didn't understand how to listen to regulators. Now that we're seeing part of that group that's come in, as I mentioned before, all the different expertise, are real regulatory people who actually know how to listen to FDA and understand what they're saying, and to EMA and to PMDA. And it's accelerating like crazy. But to answer your question about what's driving what, I think the transformative nature of these therapies, the promise, drove them to create the right framework to allow us to do the work we do. But the fact that now the geographic expansion of these. We're growing in each geography, and it's spilling over into all other geographies. The regulatory agencies are way ahead.
Yeah.
They're talking to each other. They're working it out.
Which is amazing.
It is
Actually, when you think about it,
remarkable.
it's remarkable.
It's remarkable. And the framework is not that different, and we sit in front of all of them. It's not that different from place to place.
Yeah.
So, I mean, you think about that change, but then let's go back to, cell characterization and the challenges. So you have the new regulations, but
Yeah.
but you have to, drive that standardization. I mean, Carolyn, can we really, have identity, purity, and potency nailed down for this type of a product?
I think eventually you'll have it defined enough to say that your final product will work in the appropriate patient population. Yes. Will it ever be 100%? Do we have it completely characterized? I would tend to agree with you. There's always going to be a little bit that maybe we don't know. But I do think that with the advancements that we have on omics characterizations, with the advancements that we have in data analytics, looking for those correlative properties, I do think that we'll be able to say, yes, this is your product, these are your key attributes that you need to look for, and oh, by the way, these are the attributes that you would need to look for in your patient to make sure that those two things are compatible and you're going to achieve the efficacy that you're looking for.
I don't think it's a regulatory question, really. I think
That's really- yeah, it's
the
not.
regulators have been really good about allowing us to put the matrix together that we believe works. I think the question is a business question. It's a science question.
Yeah.
Is that profile that we put together
Right.
really
Does it
indicative
represent
of
our product?
mechanisms of
Absolutely,
action, right?
absolutely.
And I think the regulators have said, we get it, that we've abdicated responsibility of mechanism of action to the cell.
Yeah.
That's the whole point of the industry.
I do
Yeah.
think
is
that
we don't
caused a
know
bit
what
of
they're doing.
confusion early on, though,
Definitely.
especially
Yeah.
with the first kinds of products that were coming to market. The release criteria had almost nothing to do with the mechanism of action.
Yeah, I mean, and I think that's
Yeah.
one of the things that we've learned in this industry.
Yes.
I mean, we saw that with MSCs. We had a whole 10 years. My PhD was in the MSC space.
I think
so
she
I'm-
was talking about MS.
I'm still very attached to MSCs, but for me, and there's certain indications which I think MSCs are certainly useful. I mean, bone cartilage applications, maybe some immunomodulatory type applications, but I think in most part, there was a trend at one point for MSCs could be the cure-all, and I think that was potentially harmful, but it was harmful from the perspective of we just didn't have a grip of what the cells were doing. And I think now where we start to see things like gene-modified cell
Yes.
therapies, ex vivo therapies, and so where we have a better handle on what the cells are doing, we know what they're meant to be doing, and we have ways of measuring them, that I think has helped drive this current trend of CAR T-cell immunotherapies and so on. But I think in addition to that, we are now starting to develop the enabling tools and technologies that can assist with that. I mean, there's a big push globally on Raman spectroscopy and using non-invasive methods to try and measure things, which I think can only be a benefit. They may not all work, but that trend and that mindset, I think, is what's important moving forward.
So is identity, purity, and potency, are they all equal, or is potency still the real challenge? Because we still are working on mechanisms of action and tying clinical outcomes
Yes.
to, product
I
attributes.
think potency and efficacy are kind of a moving target. Anything that we can do now to improve our ability to ex vivo say that a product is going to be efficacious when it gets to a patient, I think is going to be a step ahead of where we were. But I would say, yeah, that one is probably still the biggest hurdle. But ultimately, when it gets to the patient and things begin to work, you can move beyond that. Yeah, I would say that that's probably still the biggest
more data.
question.
Yeah.
Yeah,
More
it's
data,
data.
more data.
Absolutely.
And you
So-
can't get there without the scale that we're talking
That's
about.
right.
You
Right.
need the numbers of patients.
To get that data.
To get that data.
Rob, talk about taking that, characterization beyond the release from the manufacturer. I mean, it's eventually got to get to the patient.
Yep.
So
Yep.
maybe comment on that.
Yeah, and I think this is kind of leading into the comparability type of discussion as well. And I think it's important that characterization and comparability, making sure that the product is what you intend it to be at the point of fill finish. That's only halfway through the supply chain.
Right.
What effect does the supply chain have on the efficacy, the stability, the comparability of the product. There are plenty of places where things can go wrong, and maybe you don't have the same level of, in-process analytics in the supply chain to be able to detect when things go wrong. Even that last step, once the product is thawed, before it's actually administered to the patient. How can we control that? How can we make sure that every drug, wherever it's administered, is exactly the same? And I think that's one of the big challenges, that there's a huge amount of effort in manufacturing. I think the same amount of effort needs to go into making sure that the consistency and the quality of supply chain is there as well. And it's down to, we've seen in clinical trials, big variations, and it's down to really how the drug was finally administered to the patient. You get one rogue site where maybe the training, wasn't of the right quality.
Couldn't agree more.
And
One
that
of the early
yeah.
mesenchymal stem cell product companies, the CEO once told me that, because we were having this discussion about clinical sites. My experience came out of the clinical sites, so I understood what clinical sites were. And it was frightening, right?
Yeah.
There were different ways of thawing, there's different ways of storing, there's different, it just on and on and on. There's not the same training. And it struck me that we were building at the time this concept of GMP, and because we didn't do that, right? So we're doing all of that, and we're controlling the manufacturing process. And I mentioned to the CEO, I said, what are we doing from the time it's getting to the patient, and how do we know that it's efficacious? We went to FDA.
Yeah.
Is what I heard. And FDA told us the final formulation was in the freezer.
That's right.
Period, end of story.
After
and that
that,
made
yeah.
that company
Yeah.
very happy. They ended up failing a trial.
Yeah.
Wow.
And I still to this day believe part of that. I mean, there's a lot of reasons why in early MSC days struggled. It was one of the first, right? So we learned a lot from those experiences.
But I completely agree. I mean, I think it's almost considered non-fashionable to look at things like freezing, thawing, but for me, this is fundamentally still part of your product.
is.
I mean, you could have the perfect manufacturing
It's an
process.
extension of your manufacturing.
Absolutely,
Right.
and if anything, it's the one that's most susceptible to risk. I mean, in terms of it's out of the manufacturer's
The controlled
hands.
environment, that's right,
Absolutely,
yeah.
the control environment, and so from my perspective, those are the areas that are most susceptible to potential contaminations or issues at the delivery site. And again, that applies to the beginning. The isolation of the cellular
Right.
material. And,
Yeah.
Well-
there's variation in isolation techniques and methods and so on and so forth. And ultimately, as we start to move towards potentially a decentralized manufacturing process where we have multiple sites around the world, we have to show we can make the same product at different
Right,
locations.
over and over.
Or
Right.
almost
Well-
the same, good enough, the
Absolutely.
same
Yeah.
product.
Yeah.
I mean,
Exactly.
it's never going to be exactly,
within the same-
exactly the same.
within the
but-
same
yeah.
specification.
Without those
Yeah, with
final
its
kind of
specification.
potency assays, it's very hard to even compare a pre-cryo and a post-cryo product to
Absolutely.
say that they are the same thing. We can show that it's stable, quote unquote, for a certain period of time after thawing based off of, viability and some other, very low-level metrics. But is it still potent and efficacious? And I think that that's a critical question.
Qasim, your degree work did tremendous work in looking at growing large amounts of cells, right, from very few cells to many, many cells, and you broke new ground on that. I think where the engineering challenge comes in, and this may sound like a plug for the biologists, and it is that we just don't know at what phase those cells stop behaving the way
Absolutely.
they behave. And there's a lot of evidence that 3 passages for an MSC is
Yeah.
about all you can get.
Exactly.
And they lose their potency. There's so many mutations per transfer, and there's half-lives.
And we're seeing
things
with
that
T
happen.
cells. I mean, T cells
Yeah.
start to
Same with T
differentiate,
cell.
and you lose the phenotype that you'd like to experience.
So it's not biology, it's not engineering, it's this beautiful combination
Right.
where the two sort of come together. That's going to solve
Absolutely.
that challenge.
But for me, and I'm going to slightly divert the conversation, but on the same topic, for me, it's a mentality issue. I mean, having come from academia where we have a big focus on research, the other big part of our activity is around the training of the next generation.
Yeah.
And one of the things for me is actually trying to get that mentality into the next generation, that there are all of these things you need to consider. Whether you're a basic researcher or translational scientist, you need to understand that your work fits into this wider process. At some point it will go through a freeze-thaw process. Just putting it into a water bath, is that a controlled process? What can you do to remove things that will introduce variation into your system. I mean, all of the work we do now, where possible, we try and move away from things like FBS and so on to defined reagents at the very least, so that we can start to do. I mean, it
Basically,
increases
it's like
the cost.
It's an
but-
8-unit operation.
Absolutely,
It's not.
absolutely.
It's not.
But it's not. But it's non-traditional training for,
It is,
for
yeah.
For an
individuals that
academic
have-
institution,
Absolutely.
that's
And
very
Focus
different.
on, yeah.
And I think
Yeah.
that's been our goal, is that we've almost had to rewire the thinking of many scientists. And I have many biologists in my group who now, I've had to try and get that translational thinking into their brains. Actually, you're not doing lab work anymore.
Right.
You're hopefully contributing to an end vision where one day this may go into a patient.
I mean, this area does seem like a great area for academic and industry collaboration. Can we comment on that? What are the opportunities in that area?
Oh, I think that they're
Besides
plentiful.
besides training, which is an
Yeah.
obvious one.
No, I think they're plentiful. I mean, so you've asked about a lot of things, a lot of changes. So one of the things that the U.S. government is doing is that they have funded now these manufacturing institutes through NIMBLE and through Army. They've also recently funded an engineering research center all focused on cell manufacturing technologies that is being led by Georgia Tech. It has 8 other affiliate universities for that exact purpose. The engineering research center that's funded by the U.S. government is to bring together academic, industry, and government, as well as clinical research. So we have the Marcus facility that is at Georgia Tech. It is utilized for training purposes. It's utilized to get these trainees to start thinking around how do you do process control, how do we reduce variability, but it's also an opportunity for clinical researchers to come in, because they do a great job and they're fantastic at getting their INDs and getting their products to trial. They do a fantastic job of managing their small GMP settings, but I think that we could significantly assist them with some research work on how you think a little bit earlier stage on scale up and commercialization. And then bringing in the industries, whether they're startup companies, whether they're tools companies that are going to try to figure out these inline sensors that we can utilize and put them in that space where they're going to need to create a product. I think there's plentiful opportunities for that, and those are some of the things that we're focused on.
And I think there's also rich expertise in some of the the analytics that have to go into this in universities, and the needs are in industry, and a lot of the expertise is there as well. But there's, I think a rich ground.
I mean,
for
and I think
collaboration.
again that's not necessarily unique to this industry, but I think one of the pleasing aspects of this industry that there has been this close relationship between industry, academia, but also these public institutions, the CCRM, the Catapult in the UK, and others, and now we have NIMBLE and so on in the US. But I mean, at UCL, we have a £10 million EPSRC manufacturing hub focused on training development, but also where we have 35 companies involved, where they provide input and feedback into our research, but at the same time, our groundbreaking, novel, blue science type research, try and push that to industry. Say, well, actually, does this, solve a challenge, that we've identified, and would it assist you in the work that you're doing? And from my perspective, that interplay between industry and academia, public research bodies, and the regulators, all of that feeds into, I think, improving the dialogue, the harmonization, and I think that has expedited, and I've seen, and that's again the clinical work we've seen kind of integrate very nicely into that, and we're now, and it goes back to this issue, we're moving away from the clinicians running this or the biologists running this now to the manufacturing challenges. This is what we're going to solve next.
So if we assume that we can do, all the characterization we need, and we're training people, are there any other issues for kind of driving the right kind of comparability in these products? Is there anything else that needs to be tackled?
I do think that going back to a comment earlier is the number of patients receiving these. So in order to really get to a defined product, if we can characterize it, your N has to get larger. You have to have enough data to support that, and that's going to have to happen over time. So I do think with these products in particular that post-market surveillance becomes very important, and perhaps even registries of what was the characteristics of that product, how are they performing over time. I mean, I think Japan has had to do a good job addressing that with their changes in their regulatory pathways. And I'm interested to see what comes out of the other governments in order to address that fact.
That's exactly the point, and that's the Japanese system is to let these medicines through if they show they're safe. Keep a very careful eye, because the trials are not big enough to purely show safety. So they show safety and some degree of efficacy. They get out onto the market, and then 7 years of
just collect the data.
post-market surveillance
getting the
and data
data
collection. Very important.
So, does it also mean that we need to characterize the patients better as well?
Yeah, I think
I would love
stratifying,
that.
I think stratifying patient populations,
yeah.
and for me, it is a data question now, and
Yeah.
I think this is why, again, learning from other industries, this term big data, bringing that in where not just on the product characterization, not just on the analytics, but understanding patient profiles, demographics, and so on, response to therapies. We're seeing some great clinical data from CAR-T, but we're seeing instances where it doesn't quite work, and understanding the patient profile is part of that.
So why are we holding regenerative medicine therapies to a different standard than biologics in this regard? What do you think's happening here that's... Because what you just said, I mean, you've all looked at data, right? You get a big data room and you fill it with data, and what you end up with is a lot of noise, a tremendous amount of noise, right? Coming up with a real stratification that really ties. Patients are complex.
Yes.
Cell products we have
Are complex, yeah.
The manufacturing environment is complex, right? You throw 3 levels of complexity at something, I'm not sure I understand
I would
how
add,
you pull any information
and
out of that.
I would add another thing to that. We're, in many cases, and I think this has been to the detriment of the industry, we're treating patients who have failed most other, if not every other, treatment available. And I think we've gone for that incredibly difficult patient population where we really have to show success. And I think if we again moving forward, I think we need to start looking at patient populations where they haven't failed every other treatment, but this is an option that they can take
And
on.
that probably will happen naturally as...
I was going to
over
say,
time.
there'll presumably become a time when some of these therapies become first-line treatment. And we're not treating the really difficult, patients that don't respond, non-responsive to conventional therapy. Cell therapy will become the conventional therapy.
I was going to say, I think another challenge though is that if you have a system like this, compliance of the patients to report and participate
Oh, the
afterwards.
complexity, of course.
Yeah,
So,
in order to get that data,
why
you
I
get
get
cured and you
concerned.
just don't show up again.
I'm sure there's an answer in data, right?
Yeah.
I'm
Yeah.
a data believer. I really worry about this. So many
It's
experiments
making
that come
sense
out
of the data.
And I'm not sure that it's required, right? You're asking why are we holding ourselves to a different standard? And I'm not sure that we have to have that. I'm just thinking for the next generation of personalized medicine, the data is going to be out there. It just comes down to how do we collect it and then how do we analyze it. It'll collect over time. I think it'll be fantastic when we get there, but do we need to be there right now? No.
My sponsor, when I received my PhD, his name was Albert Gordon, and he was an old salt. And he, one of the greatest pieces of advice he ever gave me was, "You'll be judged by the questions you ask, not by the answers you give." And I think that's the key to data. Is just asking the questions that we need to know the answers to.
And you know what? What a perfect way to end this part of the conversation.